Hi, dear students. I welcome you to the course on Advanced Computational Techniques. Myself, Dr. SIVA, here from the Department of Physics and Nanotechnology, SRM Institute of Science and Technology. In this, we'll discuss about DNA computing, DNA computation in ciliates, bacteria, rhodopsin, how they are actually being useful for computation purpose. We are actually learning the actual computation process which is actually going there, and we are actually going to regulate those processes according to our convenience and our needs for problem solving. Let's discuss one by one. Okay. First one is the Adelman experiment. In this slide, we introduce Adelman's landmark experiment. He used DNA to solve the Hamiltonian path problem, which is called HPP. The problem is defined on a directed graph with a starting and end point. The question is whether there exists a path that visits every other vertex exactly once. The problem given here is NP complete, meaning it represents the most difficult type of problems faced in optimization and operation research. Most difficult type of problems faced in optimization and operations research. Their solutions are easy to verify but extremely difficult to find without checking enormous number of possibilities, because NP complete problems require exhaustive search. Because NP complete problems require exhaustive search. They are ideal candidates to test new computing methods such as molecular computing. Edelman's insight and method. In this slide, we see how Edelman approached the problem. He used DNA molecules so that their natural chemical behavior would perform most of the computation. DNA ligation and hybridization allows to join together, and Edelman used these processes to represent vertices and edges in the graph. He utilized DNA ligation and hybridization allow strands to join together, and Edelman used these processes to represent vertices and edges in the graph. Ligation and hybridization. Okay. Vertices and edges. He assigned single standard DNA sequences to represent the vertices. He assigned single standard DNA sequences to represent the vertices, and complementary sequences to represent the edges which connecting them. A small picomole quantity of each molecule was synthesized, and the chemistry allowed strands to join in every possible combination, producing longer DNA strands that represented possible paths of a graph. Detecting the solutions. Okay. In this step, Adelman used biotechnology tools to separate and detect whether a DNA strand representing a correct Hamiltonian path is existed in the solution. There will be a number of things will be actually produced in the solution. Out of that, the DNA strand which is representing the correct Hamiltonian path is existing or not, it will be actually identified. If such a strand was found, the answer to the original problem was yes. This experiment demonstrated that molecular biology techniques are now advanced enough to perform real computation, and that molecular computing should be explored seriously, not just as a theoretical data or theoretical idea. DNA computing in ciliates. Let's see here. What are all the, before we go into the DNA computing in ciliates, we will see here the results which is being produced by the adult one. One step had actually produced approximately 10 to the power 14 operations, only minus 8 kilocalorie per mole for ligation process being used, and 1 joule is sufficient for 2 into 10 to the power 19 operations. This is a theoretical limit which is actually 34 into 10 to the power 19 irreversible operations per joule, which is very close to that. Okay, 10 to the power 19, 10 to the power 19 is actually been achieved. Okay, only 234. Human's algorithm summary. First molecular computation is actually being given. Yes. Attempt to repeat the experiment was actually initially failed, but again and again he had tried and he had actually achieved somehow. There are certain complexities like a linear complexity in biosteps, exponential complexity in DNA strands. He had actually identified several issues also, but at last he could actually able to make a massive parallelism, which is actually yeah, incomparable with any other things. DNA computation in ciliates. This slide introduced different perspective by Landweber and Kari in 1998. They suggested that the origin of DNA computing may exist naturally in ciliates, single-celled organisms. They are single-celled organisms. These organisms, such as Axtericia nova and Axtericia trifolax, they appear to solve a similar problem to the Hamiltonian path problem. When they reorganize their genes during reproduction, while they reorganize themselves during the reproduction system, it solves Hamiltonian path problem. Ciliates have two nuclei. One is actually the macronucleus, that is active, and a micronucleus which is actually inactive. Macronucleus and micronucleus. But during the process, many non-coding segments must be removed and the coding sequence must be rearranged into the correct order. During the process of reproduction, many non-coding segments must be removed and the coding sequences must be rearranged into the correct order. We see the ciliates may have been performed molecular computation for millions of years. The rearrangement process is essentially the same as finding the correct sequence in a Hamiltonian path. Short repeated DNA sequences appear to act like matching tags that help order the fragments. In this mechanism is indeed based on processes similar to the Edelman's, then nature invented molecular computing long before humans did. Nature invented molecular computing long before humans did. Bacteria rhodopsin. This slide introduces bacteria rhodopsin, an older alternative to DNA for molecular and optical computing. Bacteria rhodopsin is a protein that contains rhodopsin, which is also found in the human eye. It exists in the membrane of Halobacterium holobium, an organism which lives in high salt environment. Halobacterium holobium, which is an organism, lives in high salt conditions. Because of harsh environmental exposure, this protein can switch among different atomic states extremely efficiently, thousands of times more efficient than the artificial materials. That was the extreme efficient here mentioned. We see that the bacteria rhodopsin can switch states by absorbing green or blue light, and it can do this up to 10 million times before degrading. They can switch the states by absorbing green light or blue light. It can do this 10 million times before degrading. Here you can actually see the bacterial rhodopsin, which is actually having a uh, 1, 2, 3, 4, 5, 6, 7 helical structures you can actually see here, where the retinal is actually found inside. Okay. It may become core component in molecular computer memory. Okay. However, unlike DNA-based systems, this method is more passive and does not actively produce a process logic, but it still represents an important direction in molecular computing research. So this allows us to go for light harvesting protein utilized for switching applications. Once the light falls on this, and it switches the direction, which is being useful in terms of logic switches we can utilize. So with this, we can actually stop this lecture, and we will be seeing the next lecture in the next video. Thank you, students.